Hi, in this video we'll continue discussing enzymes. In particular, we want to focus on the mechanisms whereby an enzyme will reduce the activation energy of a reaction. And then we'll end up talking a little bit about protein folding, which has some daunting conceptual difficulties. In this first slide, we see in blue an enzyme called lysozyme, which acts upon polysaccharides, which are strings of sugar, which are shown in red. The reaction cleaves a longer polysaccharide into two smaller fragments. And here you can see some of the details. You have a glycosidic bond between the two sugar molecules. You have two amino acids, glutamic acid and aspartic acid, making simultaneous attacks on the glycosidic bond, proceeding to a covalent intermediate with this sugar. And then we have the hydrolysis of a water molecule, adding a proton to the glutamic acid and putting a hydroxyl group on the carbon, cleaving this ester bond, which is an intermediate. And now you have a cleaved polysaccharide. And that's a pretty nice illustration of an enzymatic catalytic mechanism, but it's a fairly complicated one. So I'd like to help build your intuition about a kind of enzyme mechanism, and really the only rule you need to know to understand this example of catalysis is that opposite charges attract and like charges repel. This figure from the book shows three mechanisms of enzyme catalysis. The first is undeniably important. It's simply an enzyme which holds the reactants in the proper orientation near each other, giving them plenty of time to react so that they don't have to rely on fleeting interactions from collisions in solution based on diffusion. The second mechanism is the one we'll talk about more, and that is the electronic stabilization of the transition state. However, this picture is somewhat flawed as you don't see a transition state. You have a charged molecule. What you see is an electronic stabilization of the molecule itself, which is not going to give you catalysis, but we'll talk about that. And finally, I think I disagree with this third mechanism of physically squeezing or bending a molecule, but perhaps I don't understand exactly what they're talking about. But in any case, I think the consensus opinion is that this does not contribute greatly to catalysis mechanisms. So anyway, now we'll talk more about the mechanism 2, and we'll also use that to discuss some features of the protein structure in a little greater detail. First, let's start by talking a little bit more about an amino acid. In solution, we've talked about it being a zwitterion with a positively charged amino group and a negatively charged carboxyl group. And here's a peptide bond with the C-alpha carbon, another amino group here, and another carboxyl end here. If we look along these four atoms in the peptide bond, there's actually a fairly significant dipole moment where we have excess electron density at the oxygen end and a relative electron deficiency at the proton. So that results in a dipole moment of about 3.5 dBi, which is quite large. And I should just mention that for whatever reason, who knows, in biology the convention is different from physics. In biology, the dipole arrow points toward the positive end of the dipole. That's the opposite of what you may have learned in physics class. So we have big dipole moments at peptide bonds. Now I'm going to talk about those dipole moments when the amino acids are in an alpha helix. Let's look for the four atoms that we just saw in the peptide bond that defined the dipole moment, O, C, N, and H. Here is a peptide bond. We have the oxygen, the alpha carbon, the amino group, and the hydrogen. And so we have, in this case, a dipole, but it's pointing almost along the alpha helix axis. And we can see many of those. Here's another one. Here's another one. Now in this slide, it's emphasizing all the different dipole moments of the individual peptide bonds. And these individual dipole moments will sum into a fairly significant vector that points down the alpha helix. This has the effect of creating a significant electric field at either end of the alpha helix. Now let's imagine a hypothetical active site where there's going to be a molecule reacting in here. And we have four alpha helices lined up pointing towards the active site. It's not at all unusual to have this sort of an orientation, although it would be spread out in 3 dimensions rather than 2. Now, to understand a little bit more about the stabilization of a transition state, we need to think about a molecule in a generic transition state. And I've just drawn a molecule with 5 atoms here. And we're going to pretend that this enzyme is going to cleave the bond between atoms 3 and 4. Here's the starting material with a normal bond between 3 and 4. And here's the product with a broken bond between 3 and 4. In the transition state has a stretched bond that is longer than the normal bond, and it's on its way to being broken. Because you have a certain amount of electron density between atoms 3 and 4, stretching it out over a long distance means that there's going to be a less electron density in the transition state relative to the ground state. So you can think of that deficiency of an electron as being a partial positive charge appearing at the midpoint of the bond. So now we're going to place the transition state of the molecule in our hypothetical active site and think about the electrostatics of the alpha helices. So now we're seeing the dipole moments of the alpha helices, and we see that focused here on the active site where we have a partial positive charge in our molecule, we have significant partial negative charges at the end of all the alpha helices. This is going to stabilize the energy of the transition state, lowering its energy. Again, because opposite charges attract. This means that you're lowering the energy of putting a positive charge in this region. This is the way in which you will reduce the energy of the transition state, leading to an increased reaction rate. Now you have to remember that neither the starting material or the reaction product has this buildup or deficit of electron density or a buildup of partial positive charge. It's only the transition state that has this positive charge. So it's only the transition state that is stabilized by this orientation of electric fields at the active site. So while it's not the details of an enzymatic reaction mechanism, I think the generic point is so simple once you break it down that it provides some insight into how enzymes work in general. I'd just like to say a few quick words about protein folding, but because it's such a complex subject and not completely understood, I don't want to delve into it much more than that. But as we know, that proteins are really only active in their folded forms. In their unfolded forms, as we see here on the left, this is not going to be capable of doing the kinds of biochemistry that we want it to do. On the right here, we can see the folded form with an alpha helix, 2 beta pleats in the final folded form. And now, this is the active form. So the question is, how do you get from here to here? And do you think that's a challenging problem or not? Well, it turns out that it is a very challenging problem. As Cyrus Leventhal pointed out, in fact, numerically, just how challenging it really is. Imagine a protein made up of 100 amino acid residues. And for the sake of an argument, let's assume that there are 3 conformations for each amino acid. That means that the total number of conformations possible for this molecule is 3 to the 100th power, which is an astronomically large number. He then went on to postulate or calculate, assuming that it takes 100 picoseconds, which is less than a nanosecond, to convert from one conformation to another. The result is an unbelievably long time for just a single protein to sample all the different conformations, which is the only way that you can rigorously conclude that you are at the absolute global thermodynamic minimum when you're folding a protein. Contrast the results of this calculation with what's known about protein folding experimentally in vivo, which is that it can take microseconds or milliseconds for an unfolded protein to fold correctly. Clearly, the protein is not sampling all the conformations available as it's folding itself. And there's a fundamental process going on whereby it can achieve a folded state much more rapidly. This slide encapsulates much of the current thinking about protein folding. Here we have a linear chain, the primary structure. And when you have that, you very rapidly collapse local amino acids into stable structures of alpha helices and beta sheets. So it's thought that secondary structure units form very quickly, especially since it's a local interaction. And now, instead of having to sample conformations for, I don't know, maybe 15 amino acids here, we can treat this alpha helix as a single unit. And we can begin to arrange conformations of just this one unit, which drastically reduces the number of potential conformations available to the protein if we lock in a bunch of sequence as secondary structures locally with different elements. So the next thing that occurs is the cell trying out the relative orientations of the various secondary structural units. And when they achieve some stabilization, then you'll get some of these loops that are not recognizable secondary structural folds packing in around those. And then the protein will minimize the energy of conformation, settling into a fully folded state. Excuse me. The other important aspect which drives this process is that those hydrophobic residues will quickly try to seek out other hydrophobic residues to sort of coalesce around a hydrophobic core, minimizing the contact with the aqueous solvent. A term one hears frequently in protein folding discussions is the stage called the molten globule, where this linear chain has started to condense into a roughly compact form, yet it still has a greater spatial extent than the final product. So it's compact, but it's still a little larger so that it has room to move around and sample some conformations between residues. Okay, that's all I wanted to say in this video about protein folding and enzymatic catalysis. Thank you for listening.